So to begin with, hello everyone. Thank you very much for attending this workshop. My name is Keaun and I'm the CEO and founder of Neurosnap. And in this workshop, essentially what we're going to be doing is we're going to be talking about protein binders, specifically why they're important, how to design them, how to choose different modalities, as well as most importantly, how to screen and actually analyze some of the designs for binders. So in terms of how we've structured this workshop, we're going to start off with more basic concepts to build a little bit of a foundation. And then after that, what we're going to do is we're going to go into slowly more advanced and more interesting concepts and topics. So hopefully by the end of this presentation, each and every one of you will be able to apply some modern bioinformatic approaches for your different binder design workflows and pipelines. Now, before we get started, I just want to give a quick background on what Neurosnap is. So Neurosnap is an online platform where you can access a bunch of different tools and models for research in molecular biology, pharmacology, chemistry, as well as several adjacent fields. So we're currently being used by over 50,000 different scientists globally, including researchers in industry, Fortune 100, academia, and government, and many other common use cases that we support include protein folding, docking, molecular dynamics, and many more. Now let's talk about some of the motivation on why we should actually invest our time and efforts on developing protein binders as opposed to alternatives. As I'm sure many of you already know, binders are essential to modern molecular biology. One of the most obvious use cases is engineering binders for therapeutic purposes, such as in trastuzumab. And beyond that, binders have notably also been engineered as degraders for diagnostic purposes, for imaging, as well as in synthetic biology. Some notable examples include therapeutic antibodies like adalimumab, nanobody sensors, DARPin binders, miniproteins, and many other different types of modalities and use cases. Additionally, let's talk about some of the challenges in binder design. So even though in silico binder design solutions have came a long way compared to just a few years ago, there are still a number of different challenges that we need to be aware of. Specifically for binder design, it generally is quite a multifaceted problem where several properties like affinity, specificity, and depending on your use case, different developability properties need to be optimized simultaneously. Moreover, protein folding and interface geometry tend to be highly complex and are generally a lot more dynamic than we give them credit for, so that's another important consideration. And lastly, many of the solutions that we're going to be talking about today are going to have some limitations. So it's going to be very important for us to actually learn about these limitations in order to best tackle them. Now, the first thing we're going to be doing is we're going to be talking about different binder modalities and how we can optimally select them. In this presentation, we use the term modality to primarily refer to different structural formats or classes of binding proteins, noting that the definition might vary across a field. As a general rule, the optimal modality is going to depend on the intended application, as well as the advantage that each format offers and its limitations. So we'll highlight some of the most widely used modalities and discuss some of their key advantages and trade-offs. So over here, we prepared a little bit of a table that covers some of the most popular modalities that we've seen. First up is antibodies. The main advantages is that they have exceptional affinity and specificity. They're also very well characterized and support bivalent binding. Some of the limitations are that they're large, they're costly to produce, they have limited tissue penetration, and some of the most common use cases are that they excel for therapeutic and diagnostic related purposes. Next up, we have scFvs, which are kind of similar. They're smaller, more flexible, and one of the advantages, they can be expressed in E. coli and other bacterial systems. They're generally less stable, and they also have a little bit more of a propensity to aggregate compared to some of the other modalities in this table. Next up, we have one of my favorites, which is nanobodies or VHHs. And the advantages are that they're highly stable, they tend to be more soluble and easy to express, and they have fair tissue penetration as well. So some of the downsides is they have a smaller diversity space. Some of the sequences for nanobodies tend to be more conserved, and in general, they sometimes have lower affinity compared to, say, an antibody or some other different modalities. They're also a little bit more tricky to engineer, relatively speaking. Next up, we have peptide binders. So peptides are generally very easy to synthesize and modify. They have relatively better tissue penetration. And some of the downsides is they're often unstable, but if you make a macrocyclic peptide, this actually improves the stability problem, and we'll discuss that further later on. And in general, they also have lower specificity compared to some of the other options present. And then finally, we group DARPin and miniproteins for convenience, but they tend to be highly expressible as well as they have a very tunable binding interface, relatively speaking. However, they are generally more computationally complex to design, and especially for miniproteins, they could be less similar to natural sequences, which could have disadvantages later on. So now what we're going to do is we're actually going to go over a plot that we produce using some data from the protein-based data set that Adaptive produced. And this figure basically shows the binding strength within this data set for each modality. So as we can see, in general, nanobodies have the relatively lowest success rate within this data set, though the difference is not particularly large. And scFvs and peptides actually perform overall the best for their sample size, especially scFvs. The sample size is fairly small, but if we normalize by it, scFvs actually perform the best overall, which is an interesting observation. Additionally, using the same data, we also produce plots for expression data. And in general, most of the binders express pretty well. And, in general, miniproteins and peptides seems to have taken the lead in terms of expression, which makes sense. For nanobodies, there are a few other things that we want to take into consideration. Most notably, typically nanobodies will have pretty good stability and low intrinsic immunogenicity due to, similar VH3-like frameworks. Additionally, nanobodies are also quite modular. People have made variations that are bivalent, conjugated to radionuclides for imaging, toxins, peptides, as well as many other stuff. Additionally, nanobodies are also quite efficient when it comes to microbial systems, and they're generally easy to express. So during design, we often observe that CDR3 tends to be longer, especially compared to human VH sequences, and it also exhibits the greatest diversity overall. So compared to the other CDRs. Additionally, CDR3 is generally considered to be the most important CDR to design, as it tends to be longer, so it can grab the target better, and sometimes it can access more recessed or cryptic epitopes. Moreover, nanobodies can and frequently do bind and form interfaces via side-on docking, which will make some framework-mediated contacts. Now, this is generally something that's been widely observed, and funny enough, even the Baker Lab has actually noted this within their RFantibody repository. And when you do see this, it's not a bad thing at all. It's generally quite common. Next up, we have antibodies. So antibodies are excellent for mediating effector function, including complement activation, engagement of immune cells like natural killer cells, as well as macrophages. They also tend to have relatively higher affinity and specificity thanks to a dual domain VHVL paratopes. Moreover, they've also been extensively validated clinically, and they do benefit from a much richer and much more established ecosystem of tools and platforms compared to some of the other modalities in this presentation. So the trade-off is that they're harder to design and can run into problems like chain mispairing, aggregation, poor tumor penetration, as well as slower tissue diffusion. They're also tougher to manufacture, mainly because they require multi-chain assembly and proper glycosylation. Can't really put them into bacterial systems, unfortunately. In terms of binder developability, we're going to be talking about how drug-like a molecule is, covering not just the biophysical properties, but also manufacturability, formulation stability, as well as the overall suitability as a therapeutic or for other production use cases. Now, in this presentation, the main properties that we're going to be discussing are going to be affinity, specificity, solubility, aggregation risk, expression, stability, and immunogenicity to judge whether the binder can realistically progress to a therapeutic or production environment. It's also important to note that there are many other properties that do exist and do fall under the developability property. So for instance, viscosity at different concentrations could be one example as well, but we're just going to focus mostly on these. For affinity, we have a couple different tools that can be used to actually estimate this value. So the first one and the easiest one to start with is going to be Prodigy. It basically uses different interface features like contacts, bonds, etc. to estimate affinity. It's very good for quickly filtering out poor designs, especially because it's quite fast, but it's not super reliable for more detailed ranking. So it's more so for getting rid of some of the garbage early on. Next up, we have Evo EF2. So it offers Rosetta-like predictions for both stability and binding. And when we benchmarked it, we actually found the performance to be comparable to Rosetta, sometimes even better. And the best part is it's commercially available, making it a lot more practical for industry. So definitely a great tool to keep on your radar. Next up is Stab, DDG / IMAPEP. These two can predict mutational effects on binding as well as estimate the binding probability. This can also be followed by GROMACS with binding energetic calculations, so stuff like MMP-BSA or MMG-BSA calculations, which we'll discuss later on. So generally, these are going to be more accurate, some of the fast predictors that are available, but they're going to be also significantly slower just because you're going to be actually simulating this within a realistic and physical system, which is an advantage in itself. Next up, we have the different protein folding models. So on Neurosnap, if you use a protein folding model like Boltz2, Chai, AlphaFold, etc., and Proteinics, we add a different metric called min-IPSAE, as well as a couple other different metrics that we'll discuss later on. Min-IPSAE is very correlated with affinity. So it's one of the best metrics that one could use. And it's also very cheap to calculate. It's very fast and inexpensive to do so. So we're going to talk more about min-IPSAE, but definitely one of the most important metrics to keep on your radar. Now, for the different metrics that are related to yield, NetSol P1 provides numerical solubility predictions. It ranks them predicts a value between 0 and 1 for this. AgriScan 3D can predict aggregation propensity as well as identify different regions that could be at risk. And then finally, for expression optimization tools, we offer a DNA chisel, codon transformer, and TI signer, which can be used to optimize sequences for expression within like a specific host. Now, for stability, there are a few different types to be aware of, namely thermostability, solvent or pH stability, and a general fold stability as well. For thermostability, TempSaPro is a very solid and fast model for this. It's generally very good, especially because it can predict different thermostability at different temperatures as well. So it's quite insightful if you want to specifically optimize for that property. Additionally, for general fold stability, there's Evo EF2, as well as another tool that we offer on Neurosnap with the incredibly creative name of protein fold stability prediction. Lastly, for thermo-MPNN and protein MPNN DDG, you can use them to essentially optimize thermostability and fold stability respectively. Now, if you want to predict human immunogenicity, we suggest trying deep immuno for peptides. HuMatch is also available for estimating the humanicity for antibody sequences, though the utility of humanization is kind of a topic of debate with some groups that are really advocating for it and others that may not think is necessary. So that's something to consider as well. And let me just prepare something else. All right, perfect. Now what we're going to be doing is we're going to be getting into the real bread and butter of this presentation. Most notably, we're going to be discussing how to actually succeed in different bind design methods and how we can actually identify their limitations. So for the sake of convenience, what we're going to be doing is we're going to be breaking this down into 4 different steps. So first is going to be binder generation, followed by binder screening, wet lab validation, and optionally refinement as well as affinity maturation if needed. So first up is going to be binder generation. Now, there's a lot of different binder design algorithms, and the main ones that we're going to be focusing on are some of the most popular ones that we've seen. So we prepared this table over here. The first one is going to be our very own NeuroBind model, and the nice thing about it is that it supports virtually all protein modalities. It also supports affinity maturation, which some other tools do not. And additionally, it also optimizes various properties simultaneously, and to our knowledge, this is the only publicly available tool that does this. Now, the downside is it is quite slow and it is quite resource intensive, especially because it's going to be optimizing not just affinity, but also stability, solubility, as well as some other stuff like immunogenicity for antibodies. So next up, we also have RFdiffusion and RFantibody. There's also a couple other new RFdiffusion versions that came out. So RFdiffusion 3 was recently made available, and RFdiffusion 2 was also made available, though it's more geared towards enzymes. It was made available not too recently. Fairly recently. Now, for peptides, RFdiffusion is generally a very solid approach. And if you want to design like a Fab or a nanobody, RFantibody is also an excellent option. Technically, you can also use RFdiffusion to generate different types of protein binders. So, for instance, what you can do is you can diffuse part of a scaffold, or what you can do is you can impate part of an existing template structure. So the advantages for this is generally, RFdiffusion is quite fast, and it can also be extensively configured via its context system. However, the downside is the context system is also quite convoluted. It's difficult to use, in my opinion, and RFdiffusion does require extensive downstream filtering, especially because it only outputs a backbone. So what you have to do is you also have to take the backbone from RFdiffusion, feed it into an inverse folding model like ligand MPNN, and then after you feed it to ligand MPNN, you'll have to repredict that structure using AlphaFold 2 or AlphaFold 3 or another protein folding or co-folding model. So next up is Bindcraft. Now, Bindcraft currently only supports peptides, but when it comes to actually designing peptides, the quality is very, very good for cases where it succeeds. The major limitation of Bindcraft is that it's extremely slow and it can also be quite unreliable. So in our experience, what we've seen the success rate for Bindcraft to be around 5 to 10%. So in most cases, when you run Bindcraft, it kind of fails to converge on realistic designs within a realistic amount of, within, an acceptable amount of time. So for instance, 24 hours on an A40 GPU. So it might not be the best approach for some methods, especially if you want to design, a modality that isn't a peptide. There are probably better alternatives. BoltzGen is a newer model, and it's exciting because it can support all different protein modalities, as well as some other modalities like small molecules and whatnot. So in general, my apologies, coming over a little bit of a cold, but in general, it's very fast and it's also quite configurable. And additionally, it's a little bit less established and reviewed since, compared to some of the other tools, since it's fairly new, relatively speaking. But we actually have some benchmarks from it, and it does perform quite well. So definitely a very exciting model to keep on your radar. And then finally, there's going to be custom ligand MPNN and protein MPNN based pipelines. So the nice thing is, with ligand MPNN, you basically support all different modalities. You can support proteins, small molecules, even nucleotides. And one of the major advantages is that this is extremely powerful. It's highly configurable, and it's also very accurate, especially with adequate filters. So definitely, very useful. But the downside is that this generally also makes it much more complex to use, and it might be more tedious as well for filtering review. And you'd ideally want to automate this, so maybe not as friendly for the less technical users in our workshop. And then next up, we have macrocyclic generation. So macrocyclic proteins are peptide-based structures where the N and C termini are essentially linked to form like a closed ring. And technically, they don't have to be peptides, but for the context of this protein, we're mostly going to be, talking about peptides. And now, generally, why you'd want to do this. The main thing is that this has been observed to improve stability. It's also gained a lot of traction as a therapeutic modality, so it has a couple different advantages in that category as well. And these terminal-terminal bonds can actually sometimes form naturally during protein expression. So you don't actually have to add any extra enzymes or different chemical steps. Additionally, there's a lot of different modern design tools that can support this. So there's Boltz2, there's RFdiffusion, there is AFSIC design. And generally, these are actually pretty effective at generating these macrocyclic protein structures. Just keep in mind that if you use Boltz2 or RFdiffusion, you're going to want to pair with ligand MPNN, so you can actually inverse fold the structure and get some meaningful sequences. Now, this over here is probably one of the most important plots within the entire presentation. And essentially, it tells us the experimental affinity by the different methods available within the data set that we evaluated. So it's the same data set that we talked about previously. And as we can see that Bindcraft and RFdiffusion, they perform quite similarly. Bindcraft does take a little bit more of a lead compared to RFdiffusion. And BoltzGen actually performs better than all of them. And this is mostly because even though it has a little bit less in terms of actual binders, the number of binders that are actually strong in terms of binding strength is significantly higher. And ProTRL is also a really great option. Additionally, you will notice that some entries like antibodies, they look phenomenal, right? Where they have like excellent binding overall, and there's no binders that don't, bind. However, when we actually look at the sample size, we can see that, the sample size is unfortunately a little bit small to really draw, a more meaningful conclusion. And the ones that really have sufficient sample sizes are going to be like BoltzGen, Bindcraft, ProTRL, and RFdiffusion. So additionally, another cool observation is that most of these methods actually do really good in terms of expressing proteins and creating proteins that are expressible. So that's another little fun fact. And, in our opinion, we think that developing proteins that are expressible, this is something that machine learning models are very good at. So, in general, we can consider that to be a solved problem. Next up, we also want to take a look at an example from the adaptive EGFR contest where we take a look at like Bindcraft and RFdiffusion. And Bindcraft is over here and RFdiffusion is over here. The performance is quite similar. And if we look at ligand MPNN/protein MPNN methods, they actually perform the best overall, especially if we don't include like Sorry, my apology. My headset turns off after a certain amount of time. But anyways, overall ligand MPNN and protein MPNN are really great, and it's unfortunate the other data set didn't really segregate by those methods, but definitely, something that we wanted to show. Finally, the next step that we're going to be discussing is the actual screening and filtering steps, to cover some of the designs that we produced in the previous, stage. So over here, we have a little bit of a general kind of workflow that we'd recommend to start with, and you're welcome to customize this as necessary. But in most cases, what you're going to do is you're going to want to start with, an input target structure or receptor structure, as well as optionally like a binary template or other biases like hotspots and whatnot that you can feed into the algorithm. Then what you're going to do is you're going to provide that to a model like NeuroBind or RFantibody or something like that to generate a bunch of candidate binders. These candidate binders can be sequences or they can be structures. And if they're sequences, you can just predict their structure using like Chai or boltz2. And then after that, what you want to do is you want to actually assess the quality of these different candidates. So you want to look for stuff like clashes. You want to relax the structure. You want to look for odd mutations. For instance, if you have a proline within a flexible region like a loop, then that's generally, not going to be favorable, and you're probably not going to end up with a high quality structure. So for example, protein MPNN and ligand MPNN, we've observed them to sometimes put like disruptive amino acids within flexible regions. So something to look out for sure. Additionally, you're also going to want to predict various properties. You're going to want to predict the affinity. You're going to want to see, the quality of different metrics such as LIS or min-IPSAE. And you're also going to want to review the structure, which we'll talk about later on, and check whether or not it meets your requirements in terms of developability. Once you've done this, and if you have, some decent candidates to work with, what you're going to want to do is you're going to want to feed these structures into GROMACS and perform like a quick molecular dynamic simulation, followed by ideally MMGBSA or MMPBSA for binding energetics. So what we'll do is we'll talk a little bit more about how to evaluate this stuff later on. And if you get good results from here, you can then feed it. You can then actually perform, you can actually take these to the wet lab and determine whether or not they actually bind. And then optionally, what you can do is you can also repeat this process and refine your structures as needed. So now in terms of verifying structural integrity, the first thing that we recommend is you want to automatically filter out any candidates with major clashes or stereochemical issues or even poor interface geometry. And additionally, for structures, we also recommend you use multiple AF3 models or AlphaFold 3 models. So for instance, we recommend using boltz2 and Chai1, and optionally you can also use like Protonix or something like that. There's a lot of very good options for co-folding models these days. And additionally, you want to make sure that these different models are ran in parallel and that they're consistent. So essentially, you want to kind of cross-validate the predictions from each model. As a general rule, none of these models are going to be perfectly accurate, but the probability of one of these models producing incorrect results is generally going to be much higher than multiple models producing, incorrect results. So if you take a structure and you predict it using boltz and Chai1, and in both cases you get good min-IPSAE values, and you also get very similar conformations for the binder and similar complex structures overall, and this is generally a very, very good sign that your binder is probably going to express and actually bind well, in an experimental setting. Next up, you want to also evaluate different metrics. We have a couple different metrics over here in the recommended cutoffs. Additionally, we're going to be posting this presentation later on, so you don't have to memorize this as of right now. And generally, min-IPSC is the best. LIS, IPTM, and uncertainty and P.Q are also quite good. So we want to make sure that they do meet their recommended thresholds. Additionally, as a general rule, prioritize min-IPSAE the most because it's overall the most informative. And if the other metrics are slightly lower or there's a minor inconsistency, this isn't a big deal. And the most important one by far is going to be min-IPSAE. Now, over here, we have another very, very important plot. And this is, the correlation, for between like binding strength and these different metrics. So what we've done is we've actually generated these different metrics using Chai1 and boltz. And we find that overall, Chai1 actually does a much better job at predicting structures that are realistic for, when you provide it with like a binder and your target structure. So the metric that we found to have the greatest correlation with binding was actually the min-IPSAE value generated with Chai1 when fed with an MSA generated from MMSeq2. So a lot of words there, but this is actually quite straightforward. If you use, the Chai GitHub repository, they automatically generate the MSA thanks to the MSA server. And in Neurosnap, we also automate this step as well. So this is over here for min-IPSAE. Now what we'll do is we'll shift over to LIS. So LIS also has, very high Spearman rank correlation, with binding strength. And then finally, we have IPTM, which is also performing quite well. Next up for bolts 2, we can see that the performance is a little bit worse, but Spearmint of 20 is still not bad, especially in the context of binder design. And we see that min IPSA is the best for bolts 2, followed by IPTM, followed by LIS. So now what we want to do is we want to also filter by developability. Now this step we want to ideally make as efficiently as possible. And in general, a lot of the different tools and models that can be used to filter for developability properties tend to be quite straightforward and inexpensive to run computationally, since most of these models operate in the sequence space and generally don't require a lot of compute to run. Even the ones that run on a structure space aren't particularly computationally expensive. And we also recommend automating this step. So for instance, on Neurosnap, we have a pipelines feature. This is a visual feature where you don't really need to do any programming or do anything technical. And you can essentially create like a node over here. You can supply different protein structures and you can then feed them into, all these different models. And then after that, once the pipeline is finished running, you can actually download and aggregate the results. And this is done in a way that's automatic and very easy to customize. And furthermore, for more technical use cases, we also do offer an API. Next up, we also want to evaluate the actual binding interface. So as a general rule, we can see that we want to have an interface where there's strong nonpolar contacts and ideally some degree of like electrostatic complementarity, like in this example over here. And we actually produce this coloring using Pymol with the APBS plugin. So this is quite straightforward to do. Additionally, you also want to look for structures that have lots of contacts and hydrogen bonds. So this can be easily visualized with a tool like Pymol or Chimera X. Alternatively, if you perform like a GROMACS molecular dynamics run, it will show you the hydrogen bonds over time. So this is a metric that you can actually plot and see over time. And then after, we also suggest minimizing nonpolar patches outside of the interface to avoid aggregation and off-target effects. So that's another little trick that not a lot of people know about and something you want to watch for if possible. Next up for peptides, there are a couple other considerations that we want to be aware of. Most notably for complexes, you want to make sure that they're as compact as possible. So one of the main advantages of peptides is that they're sometimes able to bind to their target and form like these longer interfaces that other modalities might not be able to do. And in these cases, try to look for structures that minimize the ROG and make very good and compact contact with your actual receptor. So for GROMACS, the other nice thing is it can show you this metric over time, which can be quite useful. And additionally, try to look for ligands that are conformationally pre-organized. So conformational pre-organized basically just means that you want to look for structures where the unbound conformation closely resembles the bound conformation. And the rationale here is that smaller conformational changes upon binding generally correspond to a lower energetic penalty for actually adopting that bound state. So this is less important, but also a nice have, if feasible. So by now, you should hopefully have a few promising candidates to work with. If not, don't worry about it too much because it's also quite common to have to repeat a lot of these prior steps a few times, especially, on the first run, not everything goes perfectly. And then if you're lucky, you'll have several strong candidates by now, which you can then proceed to do like a more deep molecular dynamics-based evaluation. So, also note that for molecular dynamics and in general for a lot of different steps in this pipeline, they're not absolute, and you are actually encouraged to play around with this because some targets are going to need a little bit of flexibility in terms of how you, create your workflow or how you create your pipeline. So for molecular dynamics, here, let me actually show you this fun little video. Essentially, if you're fortunate enough and you find multiple promising candidates, we suggest performing, a quick molecular dynamics run, followed by MMPBSA or MMGBSA for binding energetics. And this process is generally a lot more computationally expensive to run, but it can also be worthwhile for top candidates if you have the resources for it. Generally, longer simulation windows will produce more reliable results. But I suggest actually starting off with a quick 1 nanosecond simulation just so you can get a feel for the duration and computational resources that would need to be expended for a longer simulation. And personally, I also find this step to be, optional, but also very entertaining because you can take a look at the different trajectories. You can animate them directly, using our platform or using Pymol or Chimera X. And additionally, it's also quite insightful in terms of seeing like the actual dynamics of the interactions between your two proteins, or if you're targeting something like a multimer, then it's certainly going to be quite insightful. Now, in terms of metrics that you want to monitor, the main ones you want to focus on are hydrogen bonds, RMSD, RMSF, and ROG, especially for peptides. And then after, obviously, binding energetics as well. So generally, you're going to see a little bit of fluctuation at the beginning. This is quite normal, especially as your system settles from like the previous preparation stages. But overall, you'd like to see as little fluctuation as possible. So in general, once your system becomes stable, you don't want to see a whole lot of fluctuation in hydrogen bonds or RMSD. And as long as things are quite steady, this is generally indicative of, good binding or consistent binding according to molecular dynamics simulation. Now, in terms of useful resources, we offer a full API that many of our technical users can actually use for deeper integration. So we have a blog post on that with some different examples over here. We also have our aforementioned pipelines feature, which is very helpful and directly accessible from our website. So you can use that to set up more complex workflows and kind of go from there. We also have an open source and commercial friendly Python package. So for those of you who are more technical, our Python package is essentially contains like a lot of different helper functions for common use cases in structural biology as well as in general molecular biology. And some examples of what we offer is we have like an improved minimum IPSAE implementation, and we also have implementations for stuff like BSA, interface contacts, and many other stuff. It's free to use. We don't collect any data from it, and it's also a must-have for anybody that's serious about automating binder screening. Additionally, we're also planning on releasing a tool either tomorrow or later this week. So this tool will essentially help automate the entire filtration process that we discussed in stage 2. And based on the suggestions that we made and it's going to be based a lot on the suggestions that we made in this presentation. So we're going to send out an email for that later on. Now, some of the papers that we really recommend is that there are a couple different, this one over here, nanobodies and nanobody-based human heavy chain antibodies as anti-tumor therapeutics. It's a really good review, especially on nanobody biology, and it also covers a lot of different advantages of other modalities, some trade-offs and whatnot. So definitely a great paper. There's also this paper over here, predicting experimental success in de novo binder design. It's a really good meta-analysis and actually takes a look at different metrics that are correlated with binding affinity. So the main, we actually got the inspiration to, look at min IPSAE more closely thanks to this paper over here. So definitely excellent work, and we really recommend checking out their research. So now in terms of conclusion, modern binder design algorithms tend to perform pretty well, and the typical success rate for a lot of these different approaches now is reaching like 10 to 20%, with expression rates that are even above 90%. Additionally, min IPSAE is a very powerful metric, so one of the most important things you want to monitor. Another conclusion that we want to be aware of is that the actual design method is not as important as people think. The most important step is by far filtration, because as long as you're able to get a decent sample size of potential binder candidates and you're able to screen them effectively, the chances are that you are going to find something decent, just by, law of numbers and statistics. Though for, some trickier targets, obviously this number is going to be much lower. Additionally, Neurosnap is very cool, so check us out, and feel free to send us an email and whatnot if you want more information later on as well. Now, what we'll do is we'll move on to a little bit of a Q&A, and thank you all for attending this workshop as well. If you have any questions, we're happy to answer them here, and as well as later on if you want to reach us an email or message us directly. So what I'll do is I'll actually head over the Zoom section, and let me actually see the Q&A section. All right, so first question, which tool can we use to predict immunogenicity of newly designed non-natural proteins in general, not necessarily a binder? This is a great question. So we actually have a tool that we've been working on internally, and we're planning on releasing this tool in early 2026. So what I'd like to do is, if possible, just register and make an account on us, because we send an email to everybody for those types of stuff. And then after, what we'll do is, send that over to you later on. It would also be nice to discuss germinal and Chai2. For Chai2, as far as I'm aware, it's not an open source tool, so we're not really able to discuss something that we can't play around with directly. With that being said, if it does become open source, then I'd be happy to test it out and give you my own feedback on that tool. For germinal, I haven't played around with it, so I'm hesitant to comment on it. I also know that it depends on Rosetta, so because that's not commercial friendly, which makes it a little bit less of a motivation to actually play around with. Next question, can we give the recording after the seminar? Yeah, absolutely. I'm going to be posting it on YouTube, so you're more than welcome to share it. You're more than welcome to watch it later on. And yeah, okay, the other question is this, any tools for ProTax? For ProTax, actually, I do have a colleague that knows more about it. So why don't you send us an email later on, and I'll connect you with them, and they might be able to actually provide some more information. For protein affinity maturation or redesigning the interface, how good are methods like BoltzGen or RFD? For BoltzGen, I'm not sure, to be honest. For RFD3, it's fairly new. They just released the source code, so it's difficult to comment on it. I can say, though, if you use ligand MPNN pipelines, we've had a lot of customers that essentially have created these custom pipelines or makeshift pipelines where what they do is they take an existing binder structure, and then after they mutate certain parts of it that they're confident are not going to be particularly detrimental if they're modified. And then after that, what they do is they screen them using like Boltz2 and Chai, as well as the aforementioned methods within this presentation. So this is one good option for affinity maturation. NeuroBind is also a very good option for that as well. Oh, perfect. Thank you for the great presentation. Could you share the Yeah, absolutely. I'd be happy to share the presentation. Are there any good tools to model RNA protein interactions? This is a very good question. And right now, for dedicated RNA related design, unfortunately, no. However, using ligand MPNN based pipelines, there are some different ways of doing this. So, for instance, what you could do is, similar to what I said previously about ligand MPNN, you can get your complex structure featuring like, your RNA and protein chains. And after, what you could do is you can like mutate, different residues at the interface that are in contact with the RNA. You can also mutate parts of the RNA if necessary. And then what you could do is you could screen them using like Boltz2 and Chai1 as previously mentioned as well. Nice talk. Which model do you suggest if we like to generate peptides less than 300 amino acids with high hit rate? That's a great question as well. I would start with Bindcraft, just because, if Bindcraft succeeds for your use case, then your problem is probably solved, because Bindcraft is very good with peptides when it does work. However, if you are part of that 90% group where Bindcraft just didn't work for your particular structure, I would recommend trying out NeuroBind. Obviously we're a little bit biased. We love NeuroBind, but if it generally does produce good models with good min IPSA values. And then additionally, if that doesn't work, you can also try using BoltzGen and RFdiffusion. So it's a little bit of a numbers game, especially as your peptide gets a little bit smaller. But these models should be pretty decent at generating something with good metrics that we mentioned in this presentation. Thank you for the nice talk. Real cool. Oh, sorry, that moved. Where is that question? In your opinion, what is the best tool available now for small molecule and RNA binding design, peptide design? All right, so for RNA binding design, just follow the previous advice. For small molecule, that's a great question. We actually have a talk that we're going to be doing on this later on in 2026. But I would say that, probably like ligand MPNN, followed by different, more traditional molecular docking approaches. So for instance, dynamic bind is very good, and also depends on what stage you're at. Like for instance, if you want to find a small molecule that interacts with, your target, and you're in that part of like the screening process, then, there are tools like PocketFlow that are really cool because they can generate small molecules that might, be relevant within a binding pocket. Additionally, you can also take like a library of, small molecules that you suspect will interact with your target. And then you can use a tool like Dynamic Bind with Janina, which have shown promising performance in academic literature. And then also we have the the different like AlphaFold 3 like models like Boltz2 and Chai1 and whatnot, and Proteinics. Just keep in mind, if your small molecule contains stuff like irons or other transition metals, Proteinics is generally going to perform better. We've noticed that Chai1 and Boltz2 don't perform as well, but if you have the CCD code for that molecule, you can get around this. So yeah, hopefully that was useful. And if you have any follow-up questions, do feel free to send me an email later on. Next up, what are viable binder design options? Sorry, a lot of questions, so everything's moving fast. What are viable binder design options for exotic metals and ions and solutions, such as Nd3+? Where did that question move to? Sorry, my apology. The question just moved, and I have to dig around for it, including typical datasets that were used for model training. So for exotic metals, I actually don't have a good answer, unfortunately. This is not something that I've worked with at all. With that said, why don't you send me an email later on? I do know somebody else that might have some information about this. So what I can do is I can check in with them, and then after, hopefully, I'll be able to give you a better answer on that. But that's a very cool question as well. So next up, did you try using Bindcraft or BoltzGen for lead optimization and not de novo design? Personally, I haven't, but yeah, you could definitely use this for lead optimization as well. I would say if you want to actually, it kind of depends. Like some of these models, they have different advantages. In general, it doesn't really matter which generation approach you want to use. The most important stuff is going to be filtration overall, if that makes sense. Can you please provide de novo design of nanobody publication from your site? Yeah, absolutely. So we're going to be releasing a PDF version and YouTube video for this presentation. So you can just click the link once we release a PDF version of this presentation. If a protein scores highly on computational benchmarks, how likely is it to perform similarly well in a wet lab experiment? Also, can wet lab results be fed back into our pipeline to refine? Yeah, absolutely. You can definitely do this. In fact, this is something we definitely encourage as well. In terms of the actual similarity with the wet lab, so as we saw for min IPSAE with chai, the correlation is roughly 0.3, give or take a bit, depending on the target. And in general, right, that's decent enough. So it means that if you have 10 candidates where, we've seen success rates around 10 to 20%. So if you have like 10 candidates that you want to screen in a lab, for most cases, you're probably going to get at least one or two different hits. Hopefully you'll get more than that, but, it does depend a little bit on the target. But generally, these models are quite robust these days. Does the macrocyclic generation tool support conformational analysis of small molecules, peptides, or not? For small molecules like non-biopolymer molecules, currently no. But what you can do is you can just get like an SDF file or a SMILE string for those small molecules, and after feed them into like Janina or a dynamic binder, one of the co-folding models previously mentioned. And after for peptides. Yeah, I mean, for peptides, it doesn't really provide too much for conformational analysis. Usually you want to do that, after all, using one of the co-folding models that were previously mentioned. Can you talk about the different tools to generate cyclic versus linear peptides, and then also what considerations are unique for cyclic versus linear? Personally, I don't think it really changes too much if you're using a cyclic model in terms of how you're going to be screening and filtering molecules. However, with that being said, generally speaking, the most impressive tool I've seen for this is probably going to be, the ligand MPNN like pipelines. So this is where you get like a backbone or a structure that you predicted using Boltz-2 with the cyclic constraint enabled. And then after you just inverse fold it and after screen that as necessary. Are you going/planning to do some workshops on de novo enzyme? Yes, absolutely. We have another one. The next one we're going to be working on is actually, a presentation on enzyme engineering. I personally love enzyme engineering. That's what really brought me into the field to begin with. So we are definitely going to be having a nice workshop for that in probably, I want to say Q1, maybe early Q2 of 2026. And you're more than welcome to email me about this as well. If you have questions related to enzymes, I'm always happy to talk about that type of stuff. I'm curious why you mentioned that Bindcraft is primarily for peptide modalities. Where'd that question go to? I had the impression that was initially developed for miniproteins and binders. Yeah, I mean miniproteins as well. If you just extend if you just make the peptide binder longer and longer and longer, now you're just falling into miniprotein territory. But realistically, like the distinction between the two is kind of arbitrary. I guess miniproteins are also a little bit more structured, shall we say. So yeah, there's that aspect of it all, and I'm sure one could make some arguments against this, but generally, yes, you can use Bindcraft for mini proteins as well. It will just take a little bit longer because it'll increase the actual model context size for the different tools that Bindcraft uses within its pipeline. So what about the specificity? What about the specificity? Because we see a lot of backbone interactions in silico. So assuming this question is related to looking at and screening for specificity, Yeah, to be honest, I don't really understand this question, so maybe clarify this in an email later on. I'll be happy to follow up on that. What is the procedure for accessing Neurosnap for academic institutions? You can just register on our website, and then after from there, what do you call it? It's quite straightforward. You start off with a couple different credits, and after you can either do purchasing via credit card after on if you're satisfied with the product, or what you can do is you can reach out to your institution for a PO. To clarify, peptide binders, 30 amino acids, proteins. Yeah, sure. For MD simulations, is there any advantage in using GROMACS over Amber? So Amber is a force field, and with GROMACS, it supports like different Amber force fields as well. In fact, the default one is an Amber force field that I highly recommend. Additionally, it also supports like Charm and Gromos as well as OPLS. I believe it's called OPLS. So yeah, GROMACS is great. And if you want to use like another, molecular dynamics solution like OpenMM or something like that, that's perfectly fine as well. We happen to like GROMACS just because it's very fast. It's probably the fastest one overall. And additionally, if you're more into like molecular dynamics stuff and you want to use, one of the Martini force fields, it's probably a little bit easier to use GROMACS for that. But, GROMACS itself is also kind of a pain to set up. So, maybe it's not as easy as people think it is, but I don't know. Now, how many of Neurosnap's tools / integrations will be open source / something that can be run on our own computer? It's nice with the Python package and open source by playing. Yeah, so this is a great question as well. We try to balance like open source stuff as well as other stuff at the same time. So our Neurosnap tools package has a lot of stuff already that we use throughout our application. So definitely check that out. Some of the other stuff we don't open source, just on the basis that they're already open source. So for instance, right, like Bindcraft is already open source, so you can access that. Or like, Bullets 2 is an open source tool. You can access that directly if needed. With low success rate for binder prediction, what is the biggest bottleneck? Expression, solubility? I would say affinity. I would say definitely affinity, because solubility and expression are, in my opinion, like a very solved problem, as we saw in like some of the benchmarks from the protein-based data set. The expression was like 90%. So, and we've even seen it higher. Like for instance, we developed these nanobodies for another collaboration that we're doing with a different organization, and we use NeuroBind to generate these nanobodies, and we saw the expression for that one to be about 96%, if I recall correctly, and the solubility was definitely quite good too. The binding affinity was definitely the bottleneck for that one, and out of 46 different candidates, the first time we got absolutely nothing that actually bound. After the second time, when we started filtering more with my min IPSAE, we actually got much better results, and we got a couple good ones for that target that are quite satisfied with. Do you know any tool to screen hotspots for protein interactions? This is a good question as well. So there are a couple of different ways that you can do this. One option is to use a tool called ScanNet. It's available on Neurosnap, and the cool thing is it predicts binding sites within a protein as well as optionally within multimers. So you can check out ScanNet for that. And then after, for small molecules, there's also AF2Bind. Technically, AF2Bind, it also has a good correlation with ScanNet, so you can also use it for like PPIs and for like finding potential hotspots. But it is technically designed for small molecules. Good presentation for PPI, especially for AGAB. Sometimes it would bind to wrong interface. How to know that or correct it? Yeah, it's a good question. I think it would depend on the context. So if you'd like, you can send me an email later on, and I can like take a look at it and give you my opinion. These are usually just based off of, in my opinion, a lot of these are kind of like you have to handle them on a context per context basis and then just go from there. Whether Neurosnap having all tools and analysis is subscription based. So all the tool, it's based off of credits, because essentially Neurosnap's primary cost, like the main thing that we pay for, is compute and electricity. So we have a credit system. You pay for credits, and after the credits are basically deducted based off the resources that you consume directly. So yeah, it's like we have subscriptions where you get credits, but for the free tier, you get 2 free credits every month as well that you can start with and try it out. Now, are there any good tools to add glycosylation to proteins and then after proceed to run developability? Where is that question? Where did it go to? Developability assessment. as you discuss? Yeah, this is a good question as well. So for glycosylation, you could use the different AlphaFold 3-like models. So Boltz-2, Chi1, Protonix, and whatnot. You can actually add like different glycosylation sites to your protein. And then after, in terms of like, assuming you want to actually, screen that glycosylated protein in terms of developability, this is actually much harder to do because technically a lot of these tools they don't account for glycosylation sites directly. But what you can do is kind of like, a suboptimal proxy for this is you can just like take the original amino acid sequence and after feed them through like Netzel and whatnot, which will give you like not a perfect value, but it will give you an approximate that it will likely be correlated with the glycosylated version, if that makes sense. How much similarity the in silico prediction is to wet lab results? I answered that above, but as I said previously, it's about 10 to 20% in terms of success, and then after 90% for expression. So where was that question? Okay, here it is. Do you have a procedure for metal binding protein design? Depends on the metal and depends on if you're talking about like metal ions or like a small molecule that contains like, like an iron or something else like that. It also depends on the metal because not all these models are able to like deal with certain types of metals. So the best thing is like trial and error. What I'd recommend is start out with like, Boltz-2, Janina, Dynamic Bind, or Chi1. And if it succeeds, fantastic, you have nothing to worry about and you can follow the same steps that I outlined previously. And if possible, you want to try and provide everything via the CCD code. So for instance, if you have like an iron ion that you want to introduce into the system, you can use a CCD code of Fe to actually add that iron into the system. However, if it's a small molecule and you can't find a CCD code for it, then you can try Protonix. Protonix generally handles these molecules a little bit better. And, it's just a little bit of a trial and error. You can see which models will work for that particular tool based off of whether or not the tool actually runs with that input. So that's essentially the best way you can proceed, but it's a little bit more tricky. Which protein structure tool would you advise for protein peptide prediction, and which metric would you trust the most? Min-IPSA is the metric I would trust the most, followed by some combination of LIS and IPTM. And in general, for peptides, I would start with, for structure prediction, I would just start with Chi, honestly. Chi is pretty good, and we haven't been given a reason to, distrust it compared to the other ones as of yet. But, in general, too, you also want to use multiple models at the same time. So as we saw, Boltz-2, it does well, but it does underperform a bit on some cases compared to Chi. So what you'd want to do is you want to run them both at the same time. And, at the end of the day, it's kind of a numbers game as well. So what you'll want to do is you'll want to filter out different examples based off of whether or not they have like good similarity in terms of output structure and output metrics, if that makes sense. Any tool for designing functional glycosylation or other PTMs? Yeah, so this is what you'd want to use. You'd want to use like Boltz-2 or Chi or any of the other AlphaFold 3 models, as I mentioned in the previous step. I noticed the pricing page, none of the tiers include Neurofold. Yeah, so for Neurofold is specifically for enzyme design, and we offer a separate license for Neurofold. If you have any questions related to this, you can send us an email later on, and we're happy to work something out. And we're also open to collaborations for enzyme design related projects. So feel free to send me an email later on. Suggest some resources to learn binder design. This is perfect. So we actually have some, we're having like kind of like a mini course / resource that we're preparing, and we're working with like some of our partners in academia to actually, produce like a working course for this, and hopefully it'll be ready in mid to early 2026. But it's going to have a lot of information related to binder design. You can also check out the papers that I provided within this presentation. I think they're really good, especially because as you're reading those papers, you'll hopefully get more questions, and then you can kind of go down the rabbit hole of answering those questions and learning more. Can you please describe more about Neurosnap Python package and how it can be run locally? The easiest way is to just go to the GitHub page for that Neurosnap package. And then after, there's kind of like a documentation. We need to update it actually, but the best way is to probably just look at like the different modules. We document everything extensively within the actual modules. So check it out. There's a lot of cool stuff. We even have stuff for like animating proteins and whatnot, so you can create cool figures and whatnot for, impress all your colleagues with nice presentations. But yeah, and if you have any questions, definitely feel free to reach out to me as well, and I'll be happy to point you in the right direction regarding that package. The amino acid sequence of binders are quite different based on de novo binder generation tools. Yeah, this is a great question. I actually am not particularly sure because all these models for de novo related stuff, they tend to produce like sequences that have low homology to stuff we would see in like a larger like protein sequence data set like BFD or one of the data sets that MM6 uses. So I'm not really sure if there is a pattern. There probably is a little bit of a pattern because there's going to be like some overlap and some discrepancies or differences in like the training set for some of these different models. But in general, like as I said, it's a numbers game. If possible, try to use different tools at the same time for candidate generation. Then after, what you can do is you can filter them all the same way. So, this way, the nice thing about filtration is you're kind of agnostic to the binder generation algorithm that you use, which is, in my opinion, a very big advantage. Now, what would be the best tool for developing molecular glues? That's a good question as well. For molecular glues, honestly, I'm not sure. Yeah, I would have to think about that one. That's a good question. For RFdiffusion and Bindcraft take into account glycosylation? No, they don't. Unfortunately, they do not at the moment. How about using non-canonic and or L amino acids in China? Oh, unfortunately, this is not possible either. Hopefully next year, right? When Boltz 3 comes out, it'll be able to handle like L amino acids and some other cool stuff. Actually, for non-canonic, that does depend. You can do like post-translational modifications. So technically, it does handle some non-canonic or non-canonical amino acids. But those are going to be done on like kind of like a case for case basis. You can check out like the documentation page we have on Boltz 2. It's the same submission page. We have like a note in the CCD section, the PTM section. So you can go from there kind of and see if it supports the modification you'd like. Related to Bindcraft, are these 10% hit rate numbers based on the pipeline that includes MPNN polishing after design? These are based off of like what users have fed to Adaptive to get their structures validated. So, some of these like different entries that were put inside that protein-based data set, they weren't actually optimized with any specific pipeline or filtration step. So, they might have been garbage to begin with just because they didn't use good metrics. So, it's a little bit more of an unbiased data set where there wasn't really any proper filtering for all of them, though I'm sure some people probably did quite extensive filtering for their entries as well. Great talk. Oh, thank you very much. And are there any tools optimized for the design of binders where the targeted binder region is known to be highly flexible? Technically, you can use all these different tools and, hopefully you'll have some success. So for instance, with Bindcraft, RFdiffusion, NeuroBind, you can specify different hotspots, and you can specify those hotspots to include the flexible region, and hopefully the model will be able to accommodate for that flexible region. In general, in terms of like specifically for flexible region, I'm not familiarized with any specific tools for that, but just specifying hotspots should be a sufficient start. Which tool would you recommend for a mirror protein binder design? I have no idea. I have never worked with mirror proteins, so I don't know, unfortunately. Which tool is the best for de novo nanobody design and why? For nanobody design, I would probably say RFantibody, NeuroBind, and probably BoltzGen. I haven't looked at its success rate for nanobodies, but I'm pretty sure in their paper they had a couple different promising examples of nanobodies. So I would have to fact check that, but definitely those three models are an excellent start. What design tool would you recommend for designing a structure, designing a peptide based around a specific scaffold structure? For peptides, as I said previously, I think Bindcraft is the best tool to start with, and then after that you can shift over to like RFdiffusion, BoltzGen, NeuroBind, and all these other different tools. But start off with Bindcraft just because Bindcraft's whole shtick is essentially designing peptide structures. Now, are there any tools that can estimate peptide kinetics? Question 2, if I have a lot of experimental data, are there software to train fit on experimental scanning data sets for the peptide protein interaction? Okay, yeah, so this is an interesting question. For the first part of your question, for kinetics, I'd probably say GROMACS with MMPBSA/MMGBSA calculations are, the closest thing resembling that. Additionally, if your peptide is small enough, you could feed it into like bolts as a small molecule, and then after it'll actually predict like an affinity value. So that's like a little bit of a hacky way to get like you know, a rough estimate on kinetic. And then after for experimental data, you know, in terms of like actually training or fitting a model, see, that depends because it depends on like how much data you have as well as you know, what kind of data you have. So for instance, you know, as a general rule, for like deep learning models, you want like probably, you know, a larger sample size that most people aren't going to have. So, you know, some people have quoted numbers at around 10,000 entries in your data set as a minimum. Some people say it's even higher than that. Thankfully, the easiest way is honestly just take like a protein language model like ESM or like ProTrek or something like that, and then to fine tune it on, you can fine tune like an adapter head on top of the model. This is probably the easiest method to do, and it's also fairly fast as well, and you won't need as much data. Additionally, you know, if you have like mostly just categorical data as well as like you know, continuous numerical data, you can try like, you know, one of those what do you call it, like ADA boost or XG boost is, you know, a good option. So like one of those like decision tree type models are pretty easy and fast to train. Amazing talk. Which tool can we use for finding binders for disordered/protein motifs? That's a good question. You could probably use, you know, any of the above models. You know, it doesn't really matter too much. It just might be harder in general to design. So, additionally, this one's also quite context dependent. This is a good question though. So, for disordered proteins, one such example that I can think of. Actually, no, I don't think I had an example in mind, but the more I thought about it, it's probably not the best example. But you really just want to look for hotspots where, you know, on that disordered protein, there's going to be like an interaction that you can block. And additionally, if you have like a known interaction site, then generally that will probably help with the feasibility of designing around it as well. So if you don't know, then maybe you can try using like ScanNet or some other method as well to check for that. But you know, it's certainly possible with the same methods that we discussed in this presentation. I like your plug and play workflow that allows for using different flavors of different tools at different stages and further minds. Yes, thank you very much. I appreciate it. Thanks for the amazing talk. For people working on proteins that have been crystallized and have low PLDT values on AlphaFold and other sites, any suggestion on how to model its binder further and further determine its drug candidates? Yeah, that's a good question. You know, quite frankly, you can try increasing like the recycling steps. Additionally, you can also try alternative models, like for instance, maybe Boltz 2 is predicting with like low PLAT or struggling with that particular structure, but maybe, you know, Chai 1 is going to do better, or maybe Protex is going to do better, or OpenFold or AlphaFold 2 even. There's been cases where, you know, AlphaFold 2 has actually outperformed some of these AlphaFold 3 like models. So it's gonna be harder. You can try these steps, especially recycling, because you know, we've observed many people have observed this as well. That, you know, in general, if you increase the number of recycles, you're gonna end up with like much slower prediction times, but you are going to end up with, you know, a much higher quality structure in a lot of cases. Like Sergeyov Chiknov, whose last name I might have butchered just now, did a really good presentation on AlphaFold 2 and like Colab Fold and Colab Design a couple of years ago. And I remember he had like one animation where they had like you know, a transmembrane protein, I believe, that they were folding, and it took about like, I think 12 or 14 recycling steps before like the fold actually came together. But anything before that, it was just like sloppy, disordered garbage. So that's just something I would keep in mind. But, you know, in general, it kind of depends. Or use or for use or other modifications, glycans, PG, etc. in binder design. I don't really understand this question, to be honest. If you have recommendations on how to deal with target proteins that might be too large for these models or hardware, ooh, that's a good question too. So, you know, in general, you know, if you're trying to fold a protein using like Chai1 or Boltz 2 or any of these AlphaFold 3 like models, the maximum that we've been able to fold using like what do you call it, an H200, which has, you know, about 160 gigabytes worth of VRAM, was roughly like 3,500 amino acids. So that's like kind of the theoretical limit, especially if it's going to be a complex, it's going to be a little bit less than that too. So my suggestion is to probably just rent out like an H200. And if it's anything beyond that, unfortunately, it's you can try like truncating the protein, I suppose, or reducing it in a way that would allow you to fit it within the constraints of these models. But unfortunately, really big proteins are still a kind of an open problem without doing some kind of hacky solution or just getting more GPUs. There are tools for RF diffusion molecules. Sorry, there are tools to diffusion molecules controlled to get better fit in protein pocket. We're going okay, cool. How many binders is a good number to have a decent filtering? I would say probably you want at least 50. Ideally, you'd want a lot more, and it also depends on how tricky your target is. But I think 50 is, in terms of like raw outputs from some of these different models, is a good starting point. If you're doing like a custom pipeline, like you're running ligand MPNN, using some kind of different design method, probably thousands to be honest, because like even Bindcraft for like a successful run, you're generally screening like hundreds to even thousands of different candidates. I think BoltzGen does like a similar thing internally. I could be mistaken on that. And I know NeuroBind does a similar thing as well, where it screens like a lot of pseudo candidates internally before you get an output. But in terms of like raw outputs from a lot of these models, 50 is a decent starting point for RFdiffusion. In terms of backbones, maybe like 50 to 100. Some people can argue more, but we've also seen success stories with like around 50, sometimes even less. So it depends, but great question. How come for some of the binding models there's less data? Is it because they failed for other properties? It just depends on like what people submitted, because unfortunately running these experiments tends to be very time consuming and very costly. So if you have a little bit of a less established method, it might be better, but you might not have the money to prove it, which would unfortunately make it harder to convince the academic community to then prove it for you. So that's the main reason why some of these methods have less samples, at least within that data set. What is the best initial setting to design binders using Bindcraft? Can we use some initial setting for downstream filtering, for example, minimum number of H bonds at interface, etc.? Depending on like the design constraints that you're looking for, I believe the Bindcraft repository actually highlights some good examples, for like different filtering constraints. And there's also like, it depends. Like for instance, if you want to have like the beta sheet mode, then they have like a different set of filters specifically for that. You can also reach out to their authors. Their authors are quite nice, and I think they're quite helpful too. They're very nice people. If you have questions related to Bindcraft, they're definitely the right people to reach out to. Now, how to start my journey into AI for protein engineering? Yeah, that's a good question. I don't really know. You kind of have to just try stuff, to be honest. Have you ever tried to use Amber or Charm for MMGBSA instead of using GROMACS? Personally, I have not, but I'm sure it'd run basically the same, especially given that a lot of these algorithms have kind of, their implementations might vary a bit, but they're roughly the same for the most part in terms of, how the algorithm is actually written. Once you have a candidate predicted or from the lab, is there a workflow or affinity maturation of existing sequences? Yeah. This is a great question too. So for NeuroBind, what you can do is you can just feed the my apologies. You can just feed the structure you have for the candidate that you did validate, and after it'll perform like various mutations at specific sites and whatnot, and it'll try to conserve it overall. And ideally, you should get like I believe like 25 or 30 different candidates afterwards as well. That should be hopefully improved in terms of like affinity and some of the other properties the model optimizes for. Additionally, you can try using ligand MPNN based workflows. So what you can do is you can fix specific sites that you suspect might be useful to mutate. So then after that, you can generate a bunch of different variants, and then after you can screen those using some of the other methods we discussed previously and go from there. Which tool do you suggest for humanization of variable regions of murine MIBs? You can try HuMatch and see if it like supports your use case, but it might not. Beyond that, you can send me an email later on, and perhaps one of my colleagues will have a better answer for this. How would you weight IPSAE min perplexity by ESM? Perplexity by ESM is like kind of like probably a garbage metric, to be honest. I mean, it's okay actually. I shouldn't be that harsh, but I don't think it's that useful. Especially because like, ESM is a good model, but it's not the best at kind of understanding nuances related to like protein-protein interactions, at least as far as I know, as far as I've seen and tested. But min-IPSAE is definitely number one, the one I would weigh the most. And I wouldn't even really weigh them. What I would do is I would actually just do filtering. Like the way I would do it is like, for Prodigy, check for anything below some threshold. If it's below that threshold, garbage, throw it out, move on. And then after that, same thing for Perplexity with ESM. I probably lower the threshold. I probably wouldn't weigh it that much, but anything like below some threshold, just throw it out. And after for min-IPSAE, that's the most important one. Just anything like above like maybe 0.65 initially, and after you can scale that value as you need. And after you can just throw out all the garbage from there. Now, what would you expect to be the levels of success when trying target epitopes that do not have binder precedence in the training set? Um, yeah, I have no clue. I'm sure there's like cases where the models struggle quite a bit, but there's also probably a lot of cases where the model is, maybe not seeing the target epitope or may not like, have as much of rich data in its data set supposedly related to that target, but they should work fairly decently. Either way, it's quite cheap to test out. So I would say it probably doesn't matter that much. What you can do is you can just do like a couple quick, initial runs using like BoltzGen or NeuroBind or whatever. And if you get something, fantastic. You're laughing. You already have good results. But if not, then worst case scenario, you just lost like, I don't know, maybe $10 worth of compute or something. Or if you're running it locally, then probably even less. You probably just lost like maybe 3 hours of your time setting everything up. But overall, it's probably just easier to test out and figure it out. But great question. Do you have tools you recommend for testing cell penetration for design peptides? I do not, unfortunately. That is an interesting use case. If I see anything, we'll definitely add it to the platform, and we'll probably like send an email. We usually send an email once every like 2 months or something like that with all the new tools. All right, there's a couple more questions that I'm going to answer just because there's so many questions. I was not expecting this many questions. And, I really do appreciate everybody's enthusiasm actually on this presentation. So we're very glad to, see the success for this. Given the low hit rate for binder design, are there any other metrics that can be used to filter in silico hits apart from what you've used? I would say the main ones that we've tested out like a lot of different metrics actually. And like min-IPSAE really does like take the cake. EvoEF2 produces good results as well. Let me see if I can think of anything else off the top of my head. Interface contacts, hydrogen bonds are also another good one. And then after in the Neurosnap tools package, we have a metric for electrostatic complementarity. We have 3 metrics for it actually, so you can use those as weights. So if I were to, we're going to have like the neurofilter or like snap filter service coming out later on this week, which is going to have like all those different metrics kind of packaged in. But those are probably the other ones that would factor in. So like just interface stuff, because generally speaking, the more interface contacts you have, it's probably going to result in like stronger binding. And also ROG, especially for peptides, if you can get like the radius of gyration, this is very easy to compute. For peptides, it's very indicative of good and compact binding, which is correlated with binding experimentally. How much compute time and GPU resources are needed to generate peptide with complete pipeline? In terms of like the cash dollar amount, let's say if you want to rent the hardware or use like our platform, maybe like $100 or a couple $100 for most cases. If you want to actually purchase the hardware, you can get like an A40 for a bit under like 10 grand now. I think maybe even like 6 grand. You can try and find something on eBay, like an A40, and then you can run it locally. I would say if like your binder structure is something that you can run in an A40, and it's like not just your binder structure, like your entire system is something that you can like fold using like any of the AlphaFold 3 models locally on an A40, then just use an A40 or something like that. What tricks would you suggest if there are no good hydrophobic and rigid patches for hotspot bias? Yeah, that's a good question. Maybe try ScanNet or AF2Bind to see what sites they think are good binding sites. This is like very context dependent too, because if you want to like, a lot of times you want to create a binder that actually inhibits some kind of reaction. So usually you just steal the same reaction, sorry, not reaction site. What am I saying? Like binding interface, like interface formation site. So usually you can just steal like that interface site and like use those as hotspots as well. That's probably the easiest and laziest way to do it. And where was, okay, here we are. What would be the pipeline for designing a larger binder that's clamp or bowl shaped that captures the target inside? That's a great question. You'd probably want to add some kind of logical constraint where you check like the proximity of amino acids within like the peptide or binder structure, and check like the distance between those atoms and the amino acids within like the bowl or within like the cryptic pocket or whatever it is. So for instance, if they're beyond some unreasonable threshold, like let's say maybe 10 angstroms or something like that, just throwing a random number, then you can just automatically discard that. And you can also add hotspots to kind of bias the model early on into, putting structures within that region. So that's like my initial thoughts, right off the top of my head, but you could probably play around with this and refine this as well. Is the 10 to 20% success figure apply for a candidate that's passed all of the advice qual? No. These are just like random binders that random people have basically submitted to, Adaptive's platform, and then after decide to screen them. So they could have used min IPSAE or LIS or whatever, but chances are a lot of them probably didn't. So, arguably some of these methods with proper filtering, they would have had even higher success rate. And this is also something we've observed. Like, for instance, the success rate within like the first couple adaptive competitions for like RFdiffusion or Bindcraft was inherently lower than what it is today, just because we have better filtration methods. So that's like another thing to consider. The main thing is like the actual binder generation method just doesn't matter at all if you have sufficient sufficient sample size in terms of candidates that you can filter. As long as you get like good min IPSE or LIS or other metrics, then generally, the correlation with experimental success is high enough where, you will hopefully get something, fingers crossed and everything, because at the end of the day, it's still a little bit of a gamble. Thank you for the nice presentation. Do you recommend an accurate design tool for cyclic peptide design? It seems that the current tools mainly use head-to-tail cyclization, which makes the synthesis challenging. I honestly, I think like the current tools are a pretty decent start. If I see anything else, then I'd be happy to add them to the platform. As we kind of see them on a case per case basis. But yeah, that's definitely a good consideration to keep in mind. Do you have detailed tutorials for beginners on binder design with practical examples? Right now, this is what we have. We are preparing like some course material, as mentioned previously, that should provide a little bit more of an intro into this type of stuff and provide more general advice. But I think the best way to learn is just by doing. So, just go on either our website or, go, look at the different tools independently and actually try to run them and just see what you get and see how you can like optimize your process. And, really that's the best way to learn. How the Boltzgen pipeline method and RFdiffusion pipeline method, which one is better for binder design? It depends. Probably Boltzgen, based off the metrics that we saw recently. Probably Boltzgen. Thank you for this very interesting presentation. Yeah, my pleasure. Are there any tools available for designing multi-specific binders that explicitly take multiple targets into account? This is a very good question, actually. So, this is unfortunately going to go into that custom territory. We're going to probably have to use like ligand MPN or something like that. But there are some workarounds. So for instance, with like the models that accept like hotspots like NeuroBind, NeuroBind is quite decent at this actually. What you can do is you can specify hotspots on your target protein, where one region kind of interacts with like one protein, and after another one interacts with like another region, if you don't care if the interface, if it's like the same interface that interacts with like both targets. If you need to account for both targets, then this is where it gets like, sorry, if the same interface needs to bind to like two different targets, then this is where it gets a lot more tricky. You can try using like a custom ligand MPNN based pipeline where you essentially just do binder design, and after try to bias like a protein folding step later on that interacts like that particular region after you can check like different metrics for those for like both those different targets. So hopefully I explained that well, but that's one method you could do. I'm not sure how accurate it would be just because I haven't tried anything like that myself yet. Can you specify more how your prediction of developability properties is done? Sorry, a couple of questions came through and okay, here it is. Do you co-optimize affinity with developability properties? If so, how? So if we're talking about like the filtration step, sorry, my apologies. So if you're talking about the filtration step, then you just want to run like the same input into all these different models. And then after that, like NetSol will give you a value for solubility. TempStaPro will give you a value for thermal stability. And AgriScan 3D will give you a value for aggregation propensity. And then what you can do is you can like use either like cutoffs for each one, and you can just like eliminate candidates that don't meet cutoffs for each constraint. Or what you could do is you could create like a composite score that like weighs each one differently. So for instance, there was the other user who commented on this, and for developability properties, it might actually be better to make a composite score depending on your requirements, and then you can kind of just go from there. What about the specificity of peptide binders? We see a lot of backbone interactions with the binder suggesting promiscuous binding. To improve specific interactions, we need to have side chain interactions. Yeah, okay, so this is a good question. It depends on the method. I think NeuroBind does a better job at creating like side chain interactions compared to some of the other methods that I've seen. But the other thing is like a lot of these models, they kind of use like AlphaFold 2 or sorry, like AlphaFold 3, like or one of the implementations of it under the hood at some step or another for the structure prediction component. So, the thing is these models, they don't really do a fantastic job with like modeling side chains to begin with. And this is why you want to follow this like some kind of a like relaxation step. And, the best way to see like whether or not there actually are sidechain interactions that are happening and what those sidechain interactions look like is probably to do like a molecular dynamic simulation and then analyze like the sidechain contacts over time. That's probably the best step. But yeah, this is a good question. This is a very good question. What tools do you think work best for engineering CDR regions as of now? That's a good question. I think all the tools that we mentioned here do pretty decent for that. I think RFantibody and NeuroBind are probably some of the easiest to work with. And after they're also like, custom pipelines using ligand MPNN. Because what you can do for those is you can specifically kind of block out, you can specifically fix every position besides like the CDR or even like subsets of the CDR. And additionally, actually, one other cool piece of advice is we worked with another company, and what we did with them is we actually designed peptides against a target, which obviously peptides don't have CDRs. And then after what we did was we then grafted the best peptides that interacted with the target. We grafted parts of the interacting regions of those peptides and replaced them with like the CDRs of like nanobodies, and as well as like I believe an antibody or something like that. Maybe it was an scFv. And the success was pretty good. Like we got structures that had like very good binding. So that's another thing to consider. It's probably a little bit more work, but it's just another option. It's always great to have option. For all great expression results you shared, was this bacterial expression or mammalian? I don't know. It's available on the protein-based website probably. It shouldn't matter too much, but I'd imagine there's probably like mammalian expression, especially for like the antibodies and whatnot. But yeah, it shouldn't matter too much for a lot of the designs, especially for like peptides and some of the smaller structures. Anything for unbinding kinetics. Protein-based does have that data, I believe, if I'm not mistaken. I check with them. Is there any tools to predict glycan binding sites of a modeled protein? Not off the top of my head that I know of. Does the expression cell, bacteria, mammalian affect binding, and which types of cells are you using for expression? Okay, so that's kind of like the same questions before. Can you recommend any good tools for designing molecular glues? Unfortunately, I don't know enough about molecular glues to recommend tools as of right now. How to predict modifications, which is not the CCD codes. Yeah, unfortunately, this is not possible as of right now. Again, like fingers crossed, hopefully we'll have something like that as the field progresses. Maybe like AlphaFold 4 we'll be able to do something like that. You mentioned scoring thermostability and eventually pH stability. What about enzymatic stability, like lysosome and proteasomes? That's a good question. I guess it depends on like what you mean by enzymatic stability. Like for instance, there's optimal pH. For optimal pH, we have like EPHOD, which can predict that. But if you want to clarify your question and like reach out to me later on, I'd be happy to answer that question in greater detail. As an example, what resources are estimated to be necessary to produce compact peptides with suitable admit profiles? If your peptide is small enough, what you can do is you could convert it into like an SDF file or like a SMILE string. And then after we have predictors for toxicity as well as for admit, like one of them is admit AI, you can go to that section of our website. And then what you can do is you can run the tools through that. Unfortunately, for larger structures, it's not going to work out. But for smaller stuff, you can definitely do that. For someone new to protein binder design field, what would you recommend path to get in the field and where to start? Where to go from here? As I mentioned previously, just try new stuff. Personally, I don't really know. I think a lot of my own experiences came from personal interest in what was, demanded of me at a particular time. So just try out different stuff and try to explore a lot of different things, and hopefully, your fascination will kind of drive you the rest of the way. Any software for detecting differences between ancestral reconstruction for thermostability? Not off the top of my head. If I think of anything, I'll definitely try having one of my colleagues add it to our platform. Can you talk more about non-natural amino acid and D-amino acid incorporation? This is not something that I've worked with at all, and I personally, there's no obvious way that I can provide like a workflow that might be useful in that context. So, unfortunately, I just don't have the feedback as of right now to provide. Will this lecture or PowerPoint presentation, sorry, a couple of questions came in and, okay, here it is. Be revisited. Yeah, be available and revisited. Yeah, absolutely. It'll be available afterwards. What tools do you think work best for engineering CDR regions as for now? I think I answered this already, but all of them are quite good. Thank you so much for this informative presentation. Oh, my pleasure. Thank you very much. Is there any specific strategy for targeting intrinsically disordered proteins? I think I mentioned this already, but not really. I think like the general core workflow that we propose in this presentation transfers even to like these types of proteins, so there isn't too much to really consider differently. Now we are running a little bit low on time, so what I'll do is I'll take like 3 more questions or 4 more questions, and after we'll call it a day. If people have more questions, you're welcome to send me an email, and I'll get back to you all from there. The structure prediction part of the filtering process is my current bottleneck. AF2 and AF3 are somewhat slow for thousands of design. Is there an efficient structure prediction pipeline that you'd recommend? Yeah, absolutely. So generally, you don't want to be predicting thousands of structures using AlphaFold 2 or AlphaFold 3, just because like, if you put this on like an A100, for the entire complex, it's going to probably take like the better half of a week. For most stuff. Like for instance, when we were refolding all the structures using bolts 2 and Chi2 for the metrics in this presentation, it took us like about like I don't know, I think like maybe a week and a half in total for like both models. So yeah, somewhere around that ballpark. And this was on like an A100. And so in terms of A100 hours, is there like what I'd recommend is you want to ideally filter stuff before you get to that stage. So for instance, if you have a lot of sequences, right, and the model that, like for instance in the ligand MPNN pipeline, if you have like a lot of sequences, then what you'd want to do is you want to take the sequence-based models, like different developability property prediction models, like Netzel and whatnot for solubility. And if they perform very poorly on those models, the likelihood that they're going to perform very poorly in terms of structural metrics is pretty high. So for instance, like if you produce like if you take like your thousands of candidates that are in sequence form and you run them through Netzel, some are definitely going to be predicted at lower solubility. And quite frankly, if they fail that stage, they're probably just not going to make it that far, because in general, protein design models are quite good at generating proteins that are soluble. And then from there, what you can do is you can eliminate them based off of those characteristics, or you can also like generate an MSA and then have to check which positions have like disruptive mutations. So as I said in the presentation, if there's like a proline within a flexible region, then you can check for that using the primary sequence. And then after that, what you can do is you can try folding the structures using one of the models, and after eliminating them based off of like some cheap metric like Prodigy. And then from there, what you could do is you could then like, fold them again using like, another model like Boltz 2 and perform that additional validation. So it's still a little bit computationally expensive, but this will ideally eliminate some of the garbage early on. So you'll probably end up wasting like less compute. What are the best tools to design binders to MHC receptors? I think all these different tools kind of transfer to that fine. And then lastly, how to increase the affinity of a binder. I've expressed and purified my design protein and got affinity in nanomolar. How can I increase the affinity further? Yeah, okay, so if it's already nanomolar, it's gonna be a bit trickier to refine further. One option is to run the affinity maturation tools that we mentioned previously. So this could be like a NeuroBind, or this could be like a custom ligand MPNN based pipeline. You could also, try generating more versions of that binder just completely from scratch. Sometimes, you might encounter like a dead end for that particular binder, so you can just generate some other candidates. If you've already done it once, you might be able to do it again. But yeah, that's a good question. Um, and yeah, I think that's where we're gonna call it, just because I'm getting hungry at this point. But thank you all for attending, and these are honestly all very excellent questions. As I said previously, if you have any other questions, please just let me know, and we'll be more than happy to get back to you. So take care and enjoy the rest of your day.